Structure of the cell membrane. The cell membrane consists of lipids and proteins which create what is known as a plasma membrane. The plasma membrane is made up of proteins with sugars attached, cholesterol, and what is known as the phospholipid bilayer. The bilayer has hydrophilic or water-loving heads in both the interior and exterior surfaces of the layer and hydrophobic or water-feeding tails in the center of this layer. This creates a barrier to entry but allows diffusion of simple molecules. This diffusion is essential for the molecules needed for physiological change and regulation to enter the cell and waste products that could create toxicity to be moved out of the cell. The phospholipids that make up this layer are amphipathic, meaning they have regional charge differences in polarity, hence the heads being polar and hydrophilic and the tails being nonpolar and hydrophobic. Proteins are also a part of the cell membrane. There are two types, integral and peripheral. Integral proteins are embedded in the phospholipid bilayer and cannot be removed from it. Peripheral proteins are associated with the membrane surface via weak bonds but can be removed if necessary.
There are 5 main functional membrane proteins of the cell membrane that each have a unique structure that allows important functions to occur. Channel proteins are integral proteins that permit the simple diffusion of solutes across the cell membrane by providing a channel for ions and molecules to pass through. Channel proteins move molecules down their concentration gradient, not using energy. This is a form of passive transport, or the diffusion of substances via an electrochemical gradient. Ions, such as Na+, are unable to pass through the cell membrane on their own, and thus rely on channel proteins. This protein moves Na+ at a high concentration outside the cell into the cell where it is a lower concentration. Carrier proteins are the second transport protein we will discuss. Carrier proteins are integral proteins that transport molecules and ions from one side of the membrane to the other, meaning that the carrier protein is only open to one side of the membrane at a time. Carrier proteins bind specific molecules or ions at their binding site and change shape or conformation to move the molecule across the cell membrane, either into or out of the cell. Unlike channel proteins, carrier proteins may go against the concentration gradient, and thus are a form of active transport where energy, or ATP, is required. Carrier proteins can also perform facilitated diffusion, where substances are carried down their concentration gradient and no energy is employed. This is a form of passive transport of substances that do not readily cross the cell membrane. Enzymes are a third membrane protein that catalyze chemical reactions by breaking or forming covalent bonds. Substrates, or the initial reactant of the reaction, bind to the enzyme at the active site, forming an enzyme-substrate complex. Enzymes are very specific and will only allow substrates that fit the shape of the active site to bind. The enzyme may undergo a conformational change and releases the product from the active site once the reaction is complete. Receptors are the fourth membrane protein which bind to specific molecules to change permeability or metabolism. Receptors are typically transmembrane proteins which bind to signaling molecules outside the cell and transmit the signal through a sequence of molecular switches to internal signaling pathways. This sequence of events initiates a different physiological response depending on the specific type of receptor. For example, dopamine receptors bind dopamine and insulin receptors bind insulin. Structural proteins are the fifth and final membrane protein which attach to the membranes of internal cytoskeleton and join adjacent cells. Structural proteins support the cell and maintain its shape. They are frequently long and fibrous, such as keratin in hair or collagen in tendons and ligaments. Structural proteins also attach to other molecules to anchor intracellular elements to the cell membrane and create junctions between cells.
The cholesterol aspect of the cell membrane brings us to the concept of the fluid mosaic model. The model helps describe how the phospholipids within the cell membrane can move freely. Although phospholipids always keep their polar heads facing the outside surface of the layer, the model poses that proteins and phospholipids can shift past one another in the same plane. The fluidity of the cell is determined by the cholesterol to lipid composition ratio, the temperature, and the level of lipid saturation of the phospholipid bilayer. As the composition of cholesterol and temperature in the bilayer increase, the fluidity of the cell membrane increases. Due to the bent structure of unsaturated phospholipids, and increases the fluidity of the membrane as well. The structure of the plasma membrane allows for vital functions to occur such as nutrient and waste exchange, attachment to neighboring cells, and communication within the environment. The fluid mosaic model permits only certain nutrients to come into the cell through simple diffusion, such as small uncharged nonpolar molecules, and the inclusion of proteins in the membrane allows charged ions and large molecules to pass with energy, in addition to allowing unnecessary waste to exit. The cell membrane contains specializations to allow signals to be translated through both transcellular and paracellular pathways. Some of these specialized structures include the adhering belt, which attach to neighboring cells, desmosomes, which provide stronger adhesion to other cells, and communicating junctions that allow for direct communication between cells. The selectively permeable membrane and ability to receive signals from other cells allows the body to maintain homeostasis as it is faced with changing environments and allows the body to regulate itself and maintain a healthy state.